the average on the exam was 70%. Okay, so what I did was I adjusted the scores based on some of the highest scores. So everyone got 6 points added to their exam. Okay, and if you have any other questions for me, definitely let me know. Other announcements, your journal article summaries are due on Friday. Again, I have office hours today from 11 to 1, so if anyone wants to come by and talk about that, I'm available. And I think that's all the announcements I have. I posted a few animations. You guys should, take care to sit down and watch these at some point, because I've really I've previewed them, and I make sure that I post animations. Obviously, you can go on YouTube and find a bazillion animations if you want to on anything, but I preview them to make sure that they're biologically accurate, but also that they focus your attention on some really important aspects of what we've been covering. So the ones that I've posted this week are posted along this week, February 10th, the week of February 10th. And I have one on the beta adrenergic receptor signaling pathway, which you guys are all experts on now after Monday's lab. I also posted this morning 2 different action potential animations. We are going to go over action potential mechanism today. And I also posted a really nice movie that really nicely depicts the anatomy of a neuron. And these just do more justice to animations than I can really do by drawing 2-dimensional images on the board. Okay? So take time, please, and watch these. They're put there for your benefit. Okay, so today we are going to continue our discussion of the nervous system, and what we're going to be focusing on specifically is how neurons communicate down axons via action potentials. And so we're going to give a little bit of an overview of synapses and what synapses are, and then we're going to focus on actual communication down neurons. So we left on Monday talking about the different cells that make up the nervous system. There are neurons that are the cells that conduct electrical signals. They transmit information. You guys know this from Bio 101. And then much more abundant are the glial cells, which themselves do not conduct electrical impulses, but they are the supportive and nutrifying cells. Again, you know from Bio 101 that glial cells are by far the more abundant cells in the brain. But the neurons are the cells that are going to communicate via electrical impulses. Both afferent, efferent, and interneurons are going to be sending signals to communicate signals from your sensory peripheral nervous system to the central nervous system, and then efferent signals from the central nervous system to the periphery. So let's talk a little bit about how these neurons are organized. And that is just divisions of your efferent nervous system. So again, let's just quickly remind you that if you're going to go from the peripheral nervous system, that's anything outside the spinal cord and the brain. And peripheral neurons that receive sensory input are going to be integrated in the central nervous system, which is any neurons that are in the brain and the spinal cord. Right? And then from the brain and the spinal cord, you're going to send efferent signals. So this is afferent. And those efferent signals are going to be basically your output, right? How is the body going to respond to the peripheral afferent signals? So in terms of divisions of the efferent nervous system, meaning what kinds of outputs we have, there are really 2 basic divisions. There's the somatic nervous system, which mediates your voluntary actions. So really, when you think about it, the only voluntary actions you have are conducting skeletal muscle, right? Things that you can move, right? So the somatic nervous system includes motor neurons. Includes neurons that are going to innervate skeletal muscle. So that's one division. So anytime you hear somatic, think voluntary. Motor neurons. always going to mean neurons that are connected to skeletal muscle. All right, and then the other branch is vastly different, and that branch of the efferent nervous system is called the autonomic branch. And neurons that are in the autonomic nervous system mediate involuntary actions. So everything else that's not a skeletal muscle impulse, right? And there are two branches of the autonomic nervous system. There's the parasympathetic branch, and the sympathetic. So again, these are things that you guys should commit to memory. And these are just kind of basic reminders of what types of signals these mediate. So remember, we're talking about outputs of the central nervous system. And in terms of autonomic outputs, these are involuntary. You can't control them. But there are two different types of, you can kind of lump these into two categories. There are involuntary actions that are going to be more mediating your fight or flight responses, right? So kind of acute responses to save the body from danger, to flee the predator, right? Or to fight with the predator. So, or like during exercise. So those are your sympathetics. So those are going to mediate things like increasing your heart rate, increasing your blood pressure, increasing sweating, right? Those are the sympathetic responses. Those neurons mediate those types of responses. And then you have the parasympathetic neurons, which work in total opposite effect of the sympathetics. So the parasympathetics are going to provide more of your, what I call housekeeping functions. Things like lowering your heart rate, lowering your blood pressure, mediating digestion, mediating your kind of resting functions. So they work in opposite, kind of opposing action, but neither one is either on or off at any given time. It's not like you turn a switch and you have your sympathetics on and your parasympathetics off. Think of them more like a dial on the radio, a volume dial, that you have both of those neurons are firing at some basal level. But when you start exercising, the sympathetic firing is going to be turned up and the parasympathetic firing is going to be turned down. And the opposite is true. When you're sleeping, when you're digesting, the parasympathetic neurons' actions are going to be turned up and the sympathetics are going to be turned down. Yep.
Is parasympathetic, is that mediated by the medulla oblongata?
Some aspects, yes.
Like swallowing and stuff like that you can't control?
Yes, but that particular branch of the nervous system is much bigger than just that one structure. But some aspects of the parasympathetics are mediated by the medulla, yes. And we'll talk more specifically about that when we get to like respiration and other specific physiologic systems. Okay, so this image just reminds you that the central nervous system would include any neurons that are within the brain and within the spinal cord, and then you have your peripheral nervous system that has that afferent, which is your sensory nervous system, and then you also have your efferent, which we just defined as somatic motor neurons and autonomic neurons, which are sympathetic and parasympathetic. There's also a third branch of the autonomic nervous system, and that's called your enteric nervous system. We're going to talk less about this one, but the enteric nervous system kind of bridges the gap between parasympathetic, but it mediates digestion and gastrointestinal function. So your GI tract has its own branch of the nervous system, and that's called enteric. So for our discussion, we're going to kind of lump the enteric nervous system with the parasympathetic.
Doesn't that enteric, it doesn't need blood flow?
Absolutely. It's kind of a reflexive activation. But there is override from the autonomic nervous system, absolutely. This just reminds us that, so again, these are efferent neurons. Here's your somatic, an example of a somatic neuron where its cell body is in the central nervous system and its axon is going to innervate a skeletal muscle. And all this shows us is that there is only a single neuron going from the central nervous system to the effector organ in the case of the somatics. Versus in the autonomic nervous system, there's a ganglion, which is an autonomic ganglion. All that means is that there's more than one neuron that goes from the central nervous system to the effector organ. So you have this one neuron that's the pre-ganglionic neuron that's going from the central nervous system to this ganglion. That's going to synapse with another post-ganglionic neuron, and that's going to activate whatever target tissue that you've got. Smooth muscles, cardiac muscle, is involuntary, glands, the enterics, etc. So these are, again, your involuntary actions versus your voluntaries. This is just a slide to depict the difference between the parasympathetic neurons and the sympathetic. So again, we've got our somatics up here, which have a single neuron that's going to activate the effector organ. So the motor neurons, as we'll talk about when we get to the skeletal muscle system, all motor neurons release acetylcholine. So that's pretty easy. They're all cholinergic neurons. But then you have the parasympathetic neurons, which their pre-ganglionic neuron is going to release acetylcholine, as well as their post-ganglionic. So the same neurotransmitter is utilized. Versus in the sympathetics, the pre-ganglionic neuron also releases acetylcholine, but the post-ganglionic, the one that's actually going to cause that target tissue to do something, is norepinephrine or epinephrine. So you either get norepinephrine specifically from the neuron itself, or from the adrenal medulla. The adrenal medulla is activated to dump out epinephrine into the bloodstream, and then the bloodstream epinephrine can act on that effector organ. So this is just showing you the different neurotransmitters or hormones that are dumped out by the autonomic neurons versus the somatic. But you can see that acetylcholine is used in all three of these different branches as an effector neurotransmitter. All right, so we're going to specifically talk about how neurons communicate from neuron to neuron, and also from neuron to effector tissue. And neurons communicate via spaces in between neurons or in between the neuron and the target tissue, and those spaces are called synapses. So the definition of a synapse is just the extracellular space that lies between two neurons, and a single neuron that's postsynaptic to one neuron. So again, between this orange one and this green one, this would be the synapse, the space that lies between two neurons. And so one neuron can be postsynaptic to another, but presynaptic to another, right? Depending on where it is in the line. So for example, the orange neuron depicted here is presynaptic neuron, and in this case, the green would be postsynaptic after the synapse. But if you were looking at the green versus purple, the green neuron is presynaptic to the purple, which is postsynaptic, right? It just depends on where you are regarding the synapse. So this is the synapse between this orange neuron and this green neuron. This is the synapse between this purple neuron and this green neuron. And it's mostly chemical synapses that we're going to talk about, where a neurotransmitter is released into the synapse, and the neurotransmitter then acts on the postsynaptic neuron. But there are such things called electrical synapses that are mediated by gap junctions. So I'm going to very briefly talk about the electrical synapses, but we're going to mostly focus on the chemical synapses. So there are two types of synapses, and they're defined by how the neuron is communicating to the postsynaptic neuron. So the chemical synapses are thought to be the more abundant, although more and more electrical synapses are being found in the nervous system. And so the role that electrical synapses play is thought to actually be much greater than once was thought. But chemical synapses are still known to be the most commonly found type of synapse. This is when a neurotransmitter is released as a mode of communication. So again, here's the synapse between neuron 1 and neuron 2, right? This particular region right here. And in this case, the chemical mediator that's going to be released by the presynaptic neuron is a neurotransmitter, a chemical, right? And the postsynaptic neuron, in order to respond to that neurotransmitter, that chemical that's released, what is the postsynaptic neuron going to need to respond? A receptor, of course. So the postsynaptic neuron would have to have, if this neurotransmitter was, say, dopamine, in order to respond to that, the postsynaptic neuron would have to have a dopaminergic receptor, right? A dopamine receptor. So the postsynaptic neuron can actually have multiple receptor types. So for example, if there was another neuron that also synapsed on the postsynaptic neuron, say this was acetylcholine, then to respond, this neuron might have acetylcholine receptors as well. So you can have, one neuron can have multiple types of receptors and respond to multiple different presynaptic neurons. But the major point is that these are chemical signals that are activating the postsynaptic neuron. We'll talk a lot about then, now you guys are experts in cell signaling and you know that all these, kind of this chain of reactions happens once a receptor is activated. Some of these are G protein coupled receptors, some of them are ion channel linked receptors, what have you. But we're going to talk about specifically in neurons of how these signals are transduced to an electrical signal down the axon. So when the neurotransmitter binds to its receptor on the postsynaptic neuron, the response can be an excitatory response, meaning that this neuron is going to be fired and it's going to fire an action potential, or depending on the neurotransmitter, you can actually have an inhibitory response, meaning that less of a response is going to be elicited. And you can imagine in the billions of neurons that you have in your brain and in your body, that inhibitory signals might be just as important as excitatory signals, because you have this integration of signals that are being propagated based on afferent signals that are coming to the central nervous system and being integrated. There's going to be a delay because you've got the neurotransmitter has to be released into the synapse, and then that neurotransmitter's got to activate its receptor, and you've got to have that biological response. So there's actually a short delay that can be anywhere from a half millisecond to 2 milliseconds, but there's going to be a delay. We're going to contrast this with electrical synapses in the next little part of this slide. And electrical synapses are essentially going to mediate almost instantaneous signals from one neuron to the next, from the presynaptic neuron to the postsynaptic neuron. The synaptic cleft is the anatomical name for this particular space, and it's about 20 to 30 nanometers in length. So to give you some context, your typical cell is going to be 10 to 20 micrometers thick. So, and there's 1,000 micrometers there's 1,000 nanometers in a micrometer, right? And so this is a tiny, tiny space. But we're mostly giving you this to contrast to the electrical synapses, where the cleft is only 2 nanometers. So as far as the chemical synapse goes, it's 10 to 20 times wider than the electrical synapse. So the space is way, way bigger. So it's like a Grand Canyon versus the electrical synapse, where the membranes of the 2 neurons are essentially totally linked together. The electrical synapses, like I said, they're less abundant than the chemical synapses, but more and more researchers are finding that these are incredibly important in the brain. And what they tend to be is kind of clumped together, so you'll have a group of neurons that are connected to each other via electrical synapses. And what can happen with those neurons is that they can almost instantly, all of them will be activated because there is essentially no delay. And so in particular biological responses or neural responses where you have to have kind of a lawn of neurons that are activated at the same time, you'll find these electrical synapses. So electrical synapses, unlike chemical synapses, which are going to require a neurotransmitter chemical to be released into the synaptic cleft and then act on a post-synaptic receptor, you don't have that. You have cells that are connected via gap junctions. So what that would be is literally you have this tiny synaptic cleft, barely any space between the two, and there are the plasma membranes of the presynaptic and postsynaptic are joined by gap junctions. And gap junctions are essentially like ion channels, and they're going to literally connect cytoplasms. So if you have some sort of electrical signal that passes down the presynaptic neuron, that electrical signal is just going to pass right on through to the postsynaptic neuron. So do you see how they're almost together like a circuit? And so there's really no delay. And so you end up getting activation much quicker than in a chemical synapse. When we talk about cardiac physiology, some of your cardiac muscle fibers are like that too. The one, like your sinoatrial node, right, your AV node, those conducting fibers are like that too. They're connected via gap junctions, and so they don't have to wait to respond. If one fires, you get this instantaneous wave of electrical conductivity. So some of the neurons in the brain act in this fashion. So in this, because the cells are connected via gap junctions and it literally links their cytoplasms, the current that is elicited by the presynaptic neuron immediately spreads from cell to cell. There's no chemical that needs to be released, there's no receptor binding activity that has to happen, and there's no signaling cascade that needs to respond. This electrical signal just goes from one to the other instantaneously. There is no synaptic delay. These types of synapses can be modulated. It doesn't mean that they're always going to just be kind of an all or nothing, depending on the signals. So you can have other signaling receptors that are going to mediate these responses, and intracellular calcium levels or pH, membrane voltage, calmodulin. You guys remember calmodulin, which binds to calcium and activates the calmodulin-dependent kinases. Those can modify how quickly electrical signals can be passed through these synapses. And again, the synaptic cleft is much smaller. There's almost no space between the presynaptic and the postsynaptic. So again, that's all I'm really going to say about electrical synapses, although very similar types of synapses are going to be seen when we talk about conducting muscle fibers in the heart later. So the rest of the time we're going to focus on chemical synapses, which are more abundant in the nervous system, and they act via neurotransmitters. So this all sets us up to ask this question: how is an electrical signal sent down an axon? I mean, think about your nervous system. Think about the gigantic neurons that you know of, like your sciatic nerve that runs the length of your entire leg, right? So that's a bundle of neurons that are single cells, and they have these gigantic axons. The biggest axon known is a giant squid that lives in, I think, the Indian Ocean. And this squid has this hunkin' nerve that runs down the length of its body, and it's just a single nerve, but it's a bundle of axons, and these axons run a huge distance. So think of it like an Atlantic cable that has to go from Europe to the United States and back, that you need these insulated axons. But anyway, so these axons conduct chemical signals from, and they're elicited by chemicals that are released from the presynaptic neuron to the post. So how is an electrical signal sent down an axon? Across chemical synapses, not electrical, neurons communicate by transducing that chemical signal to an electrical signal, and then a chemical signal again, right? So this neuron is gonna respond to a chemical signal. That chemical signal has to be transduced via a signaling cascade. Then that's made into an electrical signal down that axon, and like I was saying with that giant squid axon or your sciatic neuron, that has to pass a huge distance depending on the neuron. So that electrical signal is transduced, and then at the end of the axon, at the axon terminal, a chemical is released again, right, into the next synapse. So it goes chemical, electrical, chemical. Obviously the electrical signal is important because you can span much further distances with an electrical signal, and rather instantaneous, as you'll learn through an action potential, which goes down the axon unidirectionally in almost an instantaneous fashion. And you can just cover more ground with an electrical signal than you can with a chemical. Okay, so this is your basic overview. A neurotransmitter that has been released by a presynaptic neuron binds to a receptor on the dendrites. Remember, the dendrites are those extensions of the plasma membrane on the sensory end of a neuron, right? There's nothing special about them. They're just extensions of the plasma membrane, and they're held out by the cytoskeleton. So the cytoskeleton has these projections, and that stretches out the plasma membrane, and that's where your receptors are going to be congregated. So the neurotransmitter receptors are hanging out there on the dendrites, and it's the synapse where you're going to have the neurotransmitter that has been released by the presynaptic neuron, and you've got your receptors on the dendrites of the postsynaptic. The binding of the neurotransmitter evokes a small change in membrane potential. So you guys now all know about resting membrane potential, right? Somebody tell me what is in most all of your cells, what's the resting membrane potential? Minus 70 millivolts. That means the inside of the cell is what? More negative than the outside, and if you took electrodes, you'd be able to measure 70 millivolts in magnitude, right? And so what happens, so that's the resting membrane potential, but when the neurotransmitter binds to its receptor, that evokes that whole receptor ligand interaction, causes cellular change such that that membrane potential is going to change. And the membrane potential is either going to get even more negative, so you're going to actually step that polarity of the membrane even more. So if it gets even more negative, that's going to be called a hyperpolarization, right? Hyper means higher, so if you make it even a stronger magnitude, like minus 90 millivolts, that would be a hyperpolarization, right? Or you could actually make that polarization less. You could actually lessen the amount of negative charge on the inside by increasing the positive charges and actually pull the difference in membrane potential smaller, and that would be called a depolarization. So we're going to be talking about depolarization, right? Polar, you guys, means like opposite, different, right? And when we say the membrane potential, we mean there's a polarization of the membrane. That the inside of the membrane and the outside are not the same. The inside of the cell is more negative than the outside, and the difference stores energy, and that energy is 70 millivolts, right? And so if I lessen that energy, that's going to be a depolarization. If I make that even bigger of a difference between the outside and the inside, meaning I'm going to make the inside even more negative than it was, that's a hyperpolarization. So get used to those terms. Hyperpolarization means you make that difference even bigger by making the inside even more negative. Depolarization is you're going to make that difference smaller because you're going to make the inside of the cell more positive than it was. So the binding of the neurotransmitter is what evokes a small change in that resting membrane potential. Either you get a hyperpolarization or a depolarization, and depending on that signal, as we'll talk about, there is an internal set point that's called the threshold. And if the threshold is surpassed, then an action potential, which is a phenomenon by which specific ion channels are going to open and close down the axon, down the membrane of the axon, and we'll learn all those steps. But if in the cell body itself, if the electrical change is enough to surpass a threshold, which is a set point set in the cell, then you'll elicit action potentials down the axon, and this is where your electrical signal will be initiated. And then once the action potential reaches the axon terminal, that is going to cause other cellular responses, including calcium rushing into the cell, and that's going to stimulate neurotransmitter release at the axon terminal. So again, you had chemical binding to the receptor, that was chemical, right? The cellular response in the cell body was electrical. You were going to change the resting membrane potential, either hyperpolarizing or depolarizing. If there was enough, it's actually depolarization that would bring us towards threshold. If you got to threshold, that would stimulate an action potential down the axon, which is an electrical signal, and that electrical signal reaches the axon terminal. That would cause a dumping of neurotransmitter into the synapse. That would be a chemical. So let's just focus on these terms that I kind of introduced briefly. Depolarization is going to mean the difference becomes smaller. Hyperpolarization means the difference is going to become bigger, right? And in terms of cells and resting membrane potential, these are just terms to describe a change in the membrane potential away from the resting membrane potential. So if our resting membrane potential is minus 70 millivolts, and I change the membrane potential to either minus 90 or plus 60, either one of those is a change away from the resting membrane potential. One of them is going to be a hyperpolarization. Those would be the ones that go lower, minus 80, minus 90, minus 100. That's a hyperpolarization. Anything that's going to be bringing the resting membrane potential more positive is going to be a depolarization. So I just have these defined here. If the membrane potential, meaning the difference between the outside and the inside of the cell, becomes more positive, meaning it's kind of going towards zero and going more positive, that's called a depolarization. So what could make that happen? So if the inside of the cell is more negative than the outside of the cell, oh, I hate this talk. The inside of the cell is more negative than the outside, and you guys know that's because of the sodium, potassium, ATPase, right? What could make the inside of the cell more positive? And you have anything at your mercy. You've got ion channels, whatever. What could I do to make the inside of the cell more positive?
Sodium ion channel.
I could open a sodium ion channel, right? And isn't the sodium driving force into the cell? So that would bring more positive charges into the cell. And suddenly the inside of the cell wouldn't be as negative with respect to the outside as it was. That is a perfect example of a depolarization, and that's actually what happens in an action potential. Is sodium channels fly open, and because of the sodium driving force that was set up by the sodium potassium ATPase, sodium comes rushing in, and it causes what's called a depolarization. Well, if your cell has a set point, like your furnace does, that, okay, I'm not gonna do anything until I depolarize to, say, minus 20 millivolts, then if you don't reach that point, then the cell's just gonna go back to normal. But if you reach that threshold, boom, like an explosive series of events is gonna happen, and you're going to cause an action potential that goes down the axon. So a depolarization would be elicited if you just bring more positive charges into the cell. Let's talk about a hyperpolarization. I normally, the cell is more negative on the inside. What could I do to make this cell more negative on the inside? What's that? Okay, you could pump out calcium, right? And that's great because you'd have to pump out calcium, right? Because calcium's higher on the outside than on the inside. Is there a positive ion that's on the inside of the cell that's higher in concentration? Potassium. So if I got rid of some of my internal positive ions, wouldn't that make the inside even more negative? So, and I've got that already set up. I know that potassium on the inside is higher in concentration than on the outside. So if I just open potassium channels, I could get potassium to rush out just based on its diffusion gradient, right? And that would cause a more negative hyperpolarization on the inside. So those are really the only two ions that you need to worry about. I mean, we know that because the powerful sodium potassium ATPase, that thing is your Niagara Falls, right? It's creating this major gradient for sodium to rush in and for potassium to rush out. And this is another example of why that enzyme is so important on your membranes, because it's going to set up this particular situation where I can depolarize or hyperpolarize if I'm a neuron, and that can cause an action potential, as we'll talk about in a few. So if resting membrane, you can just quiz yourself here. If the resting membrane potential is minus 70 millivolts, as we learned before, if I changed the potential to minus 90, what would that be? A hyper, a D, or an iso? That would be hyper, right? You're making the inside more negative with respect to the outside, absolutely. If I change the membrane potential to 0, not a trick question, but if I change the membrane potential to 0, what would that be? That would be a depolarization, right? Because I'm making the difference between the outside and the inside more similar. And that means, I'm making the difference less. I'm making them more similar, so I'm depolarizing. And then what if I change the potential to minus 50 from minus 70? Depolarization, right? Anything that's more positive than minus 70 is going to be a depolarization, and anything that's more negative is going to be a hyper, right? And now you know what mechanisms you could use to change the membrane potential. You could either open sodium channels to depolarize, or you could open potassium channels to hyperpolarize, right? And that is the mechanism that your cell uses to cause this propagation of electrical conductivity. Another thing about, one sec, Reagan. Another thing about electrical conductivity, just remember in electricity, and I'm not expecting you guys to know your circuits, although I think you guys have had physics and probably could, run circles around me regarding that. But think of electricity as charges moving. Like, electricity is going to require charges that are moving. So when sodium channels open and sodium floods into the cell, that is an electrical impulse, right? When potassium channels open and potassium leaves the cell, that is an electrical impulse. So equate these ions that are moving across membranes, equate that with the electricity, and that can kind of help you with this discussion. Yeah.
So each different type of neurons gonna have a different threshold based on like the sensitivity of these.
Okay, so each different neuron is going to have different neurotransmitter receptors. Some neurotransmitters are inhibitory, some are excitatory. So if I gave a postsynaptic neuron, if I just dumped on excitatory neurotransmitters, they're going to all summate and I'm going to get an excitatory signal. If I have some inhibitory, some excitatory, it's going to summate and possibly not reach that threshold. If I have lots of inhibitories, never going to reach
But
that
they could
threshold.
each, like the actual potential they need,
Right.
is there gonna be a different level for each type of neuron?
Yes, I think on average neurons have the same threshold,
Okay.
but I think depending on the neuron, they have slightly different membrane potential or threshold potentials.
Okay.
And the best person to ask about that would be Professor Dosey, who studies electrical impulses across neurons in her laboratory. So we use kind of like the average in these discussions. So the other thing is that like minus 70 millivolts as a resting membrane potential, that's just an average. There's some cells that have a lower or a higher in magnitude, and there's some that have lower. So it really depends on the cell type. Yeah, good question. Okay, so this is, just pretend that we're in a laboratory situation and you're not really looking at the anatomy of a neuron, but you're looking more at just a membrane of any type of cell, okay? And what you're looking at in purple is a sodium channel that's open. And you guys know because of the sodium-potassium ATPase and how it's set up its driving force, you know that if you open up a sodium ion channel, sodium is going to rush where? Into or out of? Into, right? Into? Right. So you've got this open sodium channel, and suddenly these positive charges rush in, and that's going to be bidirectionally, right? I mean, it's not like there's a wave in there or a hairdryer that's brushing some of the sodium ions to one side. This is the cytoplasm, and those sodium ions are going to just diffuse away from that spot, right? And so this is a bidirectional phenomenon. The sodium ions are going to flood into the cytoplasm, and the area of membrane depolarization is going to be localized. Way over here, some of you guys going to the bathroom a little. Go ahead. I feel like 7 people have gotten up and gone outside. Okay. So way over here on this membrane, you aren't going to get, on this side of the membrane, you're not going to have a patch of depolarization because the locality of the sodium channel opening is going to be acute. So if I had sodium channels all over the membrane that were opening and causing that sodium to rush in, then the whole membrane would be depolarized. This is an important phenomenon because what you're going to do is when that neurotransmitter signal is integrated, if threshold is reached, sodium channels are going to open locally on this part of the membrane, and that's going to cause a wave of a depolarization right there. And that's going to cause a chain of reactions. So what these sodium channels are, do you guys remember voltage-gated sodium channels? So these sodium channels are totally responsive to depolarization. So here I elicit a little patch of depolarization. That, in and of itself, opens up more sodium channels. So now sodium channels in this part of the membrane open. So now don't you get a local area of sodium coming in? And that depolarizes. Then you get the next patch depolarized because those sodium channels open, and so on and so forth. So I'm gonna show you a video that I also posted on your Moodle site, but I think it really nicely at the end kind of shows you the location of these sodium channels along the axon and shows you this wave of electrical conductivity. So let me just get to my Moodle site. And it is right here.
The fundamental task of a nerve cell is to receive, conduct, and transmit signals. Neurons propagate signals in the form of action potentials, which can travel great distances along an axon without weakening. to transmit an action potential over such a distance without weakening requires that the signal is continuously re-amplified along the way. The central molecular players in this process are the voltage-gated sodium channels, which undergo a cycle of finely choreographed conformational changes. When an action potential passes, sodium channels open in response to membrane depolarization. Sodium ions rush into the axon, further depolarizing its membrane. Within a fraction of a thousandth of a second, however, the sodium channels switch to a new inactivated state in which they are closed, but now also refractory to reopening. In this way, the membrane potential can recover quickly after an action potential has passed. The sodium channels then reconvert to the closed state, ready to be opened again when the next action potential is encountered. Let's examine the changing state of the sodium channel during an action potential. When no stimulus is present, the sodium channels remain closed, and the electrical potential measured across the membrane remains constant. However, if a depolarizing stimulus is applied by a brief pulse of electric current, the membrane will start to depolarize, away from the resting potential of about -80 millivolts. Some of the sodium channels will open, permitting sodium ions to enter the axon along their concentration gradient. If the depolarization is sufficient, even more sodium channels open, and the membrane potential rapidly approaches the equilibrium potential for sodium, about +40 millivolts. At this point, the sodium channels close, adopting the inactive conformation, where the channel is unable to open again even though the membrane potential is still depolarized. The sodium channels will remain in this inactivated state until a few milliseconds after the membrane potential returns to its initial negative value. The action potential is propagated along the length of the axon in only one direction. By examining the membrane potential and state of the sodium channels along the length of the axon, we can see why this is so. As a depolarizing stimulus reaches our section of the membrane, sodium channels open and current flows into the axon. This in turn depolarizes adjacent sections of the membrane, causing adjacent sodium channels to open, and the action potential is thus propagated along the axon. Sodium channel inactivation prevents the depolarization from spreading backward along the axon.
So, watch that on your own time, but I think that really nicely gives you an animation
This
of,
reaches our section of the
of an
membrane.
action
Sodium
potential pulsing down the membrane. And see how these positive charges behind the action potential, the sodium channels are inactive, so that patch of membrane goes back to -70 millivolts. But you're moving this electrical conductivity down the membrane totally based on these sodium channels. What's not in this video is the potassium channels that also play a major role in repolarizing the membrane. Because remember, if you want to have another action potential, like if the neuron, if your muscle needs to contract even more, right? You're lifting or whatever, you need to keep activating that muscle. You need to send more action potentials down that neuron. You can't just have one and then be done with it. And so that membrane patch behind the action potential needs to repolarize. It needs to go back to normal so that you can get a depolarization again. And so the potassium channels are instrumental in that. So what this particular slide shows you that if you didn't have the anatomical structure of the neuron as it is, if you just took a patch of membrane of any cell and you did this, you would get propagation of action potentials in either direction. If I stuck an electrode in the middle, in a resting neuron, if I stuck an electrode in the middle of the axon and I depolarized it, you would get action potentials going in both directions. But you don't, right? Because you get your signal coming from dendrites that are receiving neurotransmitter signals, an integration of that electrical signal, which is going to either surpass threshold or not. If you surpass threshold, that patch of sodium channels is going to open, and you don't have anywhere else to go behind. So the propagation of action potentials is actually always going to be in one way. But the important thing is that you understand that it's all based on sodium channels opening and potassium channels opening, And so if I were to stimulate the middle of an axon, I could actually propagate it both directions, right? Are there any questions on that? So we're gonna kind of go forward a little bit and define action potentials. So an action potential is then a series of all or nothing events. They don't change in magnitude. It's a pulse of electrical conductivity that is all or nothing. It either is going to happen or it doesn't, and it's always the same magnitude in terms of electrical impulse. And it occurs on the membrane of neurons and muscle cells. As we'll see, you have such thing as called a muscle action potential, which is absolutely essential for your skeletal muscle to be activated, right? And you have some in your smooth muscle cells and certainly in your cardiac muscle cells. So neurons are not the only cells that can conduct action potentials. Action potentials result from a very specific series of events in which certain voltage-gated channels, like the sodium and potassiums, open, close, and/or inactivate, resulting in significant changes to the membrane potential. And these changes to the resting membrane potential can elicit electrical conductivity down a membrane. So the change in membrane potential that is in one patch of the membrane spreads to the next patch of the membrane, initiating the next action potential, because now you've got fresh sodium channels that weren't open before, and now this wave of depolarization because of the rushing in of sodium ions, the next patch of membrane suddenly gets depolarized and no sodium channels open. Then you get a dumping in of sodium, and the next patch, because of diffusion, gets depolarized. Those sodium channels open, and so on and so forth. So it's this chain reaction of events. So, as I mentioned, there's distinct types of voltage-gated ion channels that are totally responsible for the action potential. So the ones that we've talked about thus far are the voltage-gated sodium channels, and they open when the membrane is depolarized past a certain threshold voltage. So you can't just go from like -70 to -69 and expect that they're going to open. There's a certain threshold at which the sodium channels will change their conformation and open. Okay? And that opening results in a huge influx of sodium into the cell, and that positive charge that leaks into the cell is called a depolarization, right? So in a way, isn't that positive feedback? A depolarization begets a depolarization, isn't that true? Right? I'm a sodium channel, I need a wave of positive charge to actually cause me to open. And then suddenly I'm going to cause more depolarization, right? So it's a positive feedback. This is actually another example of positive feedback in your body. The voltage-gated sodium channels, unlike the potassium channels that are voltage-gated that we're going to talk about, the sodium channels quickly inactivate. So they have like an extra little arm on them. So remember, the sodium channels are just proteins. They're like a 4 subunited protein that's like a pore in the membrane. But there's a little three-dimensional part to it that will automatically, once the sodium channel is open, there's a certain finite period of time, and that inactivation is called an inactivation gate. And it's just a three-dimensional structure of the sodium channel, and it'll plug up the pore. So that's totally different than opening and closing. Like the opening and closing is the pore open, the pore close. The inactivation gate, right after the sodium channel opens and sodium starts rushing in, the inactivation gate will just automatically plug it up. So why the heck is that important? Because it's very important to have that patch of membrane that just had an action potential. It's important to have that membrane in refractory, such that you can repolarize the membrane so that another action potential will be able to take place behind it. Yes?
Does the axon have to be constantly pumping sodium outside?
Yes, beautiful, right? If I'm going to maintain that driving force, the sodium potassium ATPase has to still keep that driving force going, right? And so those neurons are generating all kinds of that driving force, and so they're using all kinds of ATP, right? So remember that I said that like 40 to 60% of your ATP is generated just to fuel the ATPase, just to fuel the sodium potassium ATPase. And so this whole time, that sodium potassium ATPase pump is creating that driving force so that every time you open a sodium channel, sodium rushes in. Very good. The last The other type of voltage gated channel that's important are the delayed voltage gated potassium channels. They also open in response to a depolarization, but they're delayed. They don't open as fast. So you get depolarization, sodium channels opening, sodium rushes in, and a little millisecond or so later, the potassium channels open. They end up causing potassium to rush out, thereby bringing the action potential back to the resting membrane potential. So they open seconds after the sodium channels, actually around a second. And that opening results in a huge flux of potassium out, causing a hyperpolarization, or actually more like a repolarization. You're repolarizing the membrane potential to its resting membrane potential. So everybody, please take my advice and read the very small section in chapter 6 on